Okay, this is where we left off, and we just talked about agonist and antagonist effects of drugs, and the fact that drugs can interact with one another. So, some common drug actions. I mentioned this at the end of class, but just sort of to list some, you can act like the neurotransmitter, mimic it. You can increase production of the neurotransmitter through its precursor. You can inhibit its metabolism or breakdown. You can inhibit reuptake. You can increase the amount of neurotransmitter in vesicles. All of those things are drug effects that are agonistic. In terms of antagonistic measures, you can sit in the receptor but do nothing. That's called being a competitive antagonist. You're competing with the neurotransmitter to block that receptor. You can inhibit production. You can accelerate the metabolism or breakdown of a neurotransmitter. And you can even cause neurotransmitter to sort of leak out of the vesicles so not as much is released each time. Okay. Everything we talked about so far has been post-synaptic side, meaning actions on the receiving neuron. There are also pre-synaptic receptors that are doing some specific things. One of the most common types of presynaptic neurotransmitter receptors is called an autoreceptor. So this is a receptor we see here, norepinephrine, being released to a target, but also there's an alpha norepinephrine receptor here that is being stimulated by its own release of norepinephrine. And this provides a signal back to the axon terminal to sort of let it know this is how much you're stimulating your target. And that's a feedback mechanism. So if it's not strong enough, we might pack more neurotransmitters into vesicles. If it's too strong, we might back off on the neurotransmitter that's being released. Also important because we can design drugs that target autoreceptors and essentially trick the axon terminal into either increasing or decreasing its neurotransmitter release. The second type is a heteroreceptor, and this is where another axon can form a connection, a communication connection directly with an axon terminal and bypass the dendrites, the soma, the action potential, just go straight to the source. And this is being shown as an inhibitory one, so this is going to shut down the release of norepinephrine here when this other neuron becomes active. It can also increase it. You can facilitate or inhibit neurotransmitter output through heteroreceptors. Autoreceptors is a feedback mechanism of your own neurotransmitter release. Heteroreceptors is receiving a different neurotransmitter. All right. Neurotransmitters can be organized into classes. There's 6 basic classes. They're largely based on how each class is based on how those neurotransmitters are synthesized, on how they're created. What is the precursor molecules? How are they made? This is probably less important. What is important, though, is to understand some of the mechanisms of how neurotransmitters are created. So neurotransmitters are synthesized in a particular pathway. So let's look at acetylcholine, for example. You have choline, which is a nutrient from your diet that your liver is going to extract and get to the brain. And then you have acetyl coenzyme A, which is something that's going to be made by an acetylcholine neuron, right? Those two things, acetyl coenzyme A and choline, come together to create the chemical acetylcholine. When acetylcholine is released and does its thing, its primary way that it's broken down is an enzyme that comes in and cleaves choline apart from acetyl coenzyme A, and then that choline is recycled or reused, brought back in to be bound back to acetylcholine, into acetylcholine with acetyl coenzyme A. Hope that makes sense. Yes, that's a lot. Serotonin comes from a tryptophan, is its nutrient precursor. Tryptophan gets converted into another chemical called 5-HTP. 5-HTP gets converted into serotonin, also known as 5-HT, right? The specific like enzymes that are doing these modifications, that's not necessarily important to know unless we get to a drug that acts on one of those, right? And so sort of just understanding the system, like how that these things get created, that it's a process of enzymes modifying other chemicals to create neurotransmitters is what's important. Dopamine, norepinephrine, and epinephrine are all grouped in the same family because they all come from the same precursor. Phenylalanine in the diet gets converted into tyrosine, tyrosine converted into L-DOPA, L-DOPA converted into dopamine, and then the process stops there if it's a dopamine neuron. However, if it's a norepinephrine neuron, the neuron is going to make another enzyme called dopamine B hydroxylase that's going to convert the dopamine into norepinephrine, and then the process is going to stop. Unless it's an epinephrine neuron, in which case it's going to release another enzyme that converts norepinephrine into epinephrine. Okay. This is important because, again, one strategy for Parkinson's disease is to increase the presence of L-DOPA, therefore boosting the production of dopamine. All right, so now I want to go through some specific neurotransmitters and just sort of do an overview of them. So glutamate, traditional excitatory neurotransmitter, it gets broken down. Its primary way that it's removed from the synapse is reuptake by presynaptic neurons for recycling, okay? As I mentioned in class, many receptor subtypes. There's over 14 ionotropic, and these are just the ones that we know about. There are probably subtypes that we haven't discovered yet in the brain. At least 14 different subtypes of ionotropic. In a minute, I'll talk about the role of AMPA and NMDA glutamate receptors for memory formation in the hippocampus. There's at least 8 subtypes of metabotropic receptors. Some drugs that affect glutamate are PCP, ketamine, and alcohol. GABA. GABA is our universal inhibitory neurotransmitter. So if glutamate is the go signal, GABA is the stop signal. Its primary breakdown is reuptake again. These are our primary discrete signaling molecules to excite and inhibit other neurons. So we don't want to spend a lot of time making more glutamate and GABA. We want to recycle it as much as possible, right? 2 receptors for GABA, A and B. A opens chloride channels to allow negative chloride to come into a neuron. That's how it does its inhibitory thing. B activates potassium channels through a second messenger system. So it's actually a metabotropic receptor that activates a potassium channel. And when potassium leaves, it becomes more negative. Common drugs that affect this are pretty much anything that's a depressant or a sedative, like barbiturates, benzodiazepines, alcohol also. All right. You might say, wait a minute, I thought you said that alcohol affects glutamate. It does. It decreases glutamate and enhances GABA. So it has this antagonist-agonist effect. All right. Acetylcholine, ACH, found at the neuromuscular junction. So it is the neurotransmitter that when it's present, our muscles contract. When it's not there, our muscles don't contract. Parasympathetic nervous system, that's our return to homeostasis, slow down system. Neurons that are releasing acetylcholine, we sometimes call them cholinergic neurons. Primary breakdown, as I've already mentioned, is enzymatic breakdown. Acetylcholine esterase severs the tie between choline and acetyl coenzyme A. Lots of different receptors for acetylcholine. 2 major classes are nicotinic. This is the receptor that's found at the neuromuscular junction, the autonomic nervous system, cardiac system, in the central nervous system. It can act in the CNS to release other neurotransmitters, so it can be modulatory. And called nicotinic because that is also the receptor that nicotine can bind to have its effects. Muscarinic, mainly in the central nervous system, also found in some cardiac and smooth muscle. So smooth muscle are the muscles of our digestive system and not our axial muscles or movement muscles. And in our exocrine gland system as well, typically as a second messenger receptor. The nerve gas sarin, you may have heard about that, is being used as a biological weapon, is a acetylcholine agonist, right? And so it has its horrible effects by causing people to become paralyzed because it has high affinity and efficacy. So it's going to sit in the neuromuscular junction and cause all of your muscles at once to contract, and also affects your heart, can stop your heart, etc. Two antagonists of acetylcholine are curare, which occurs in plants, native plants in Central and South America, and so it will prevent muscles from being able to contract, so it causes paralysis that way. And indigenous people in Central and South America realized this and would rub the tips of their arrowheads on this plant to collect the curare, and then use that to paralyze animals when they were hunting. Right? Botulin toxin is another antagonist. So when you get Botox, you're actually injecting this botulin toxin into your face so that muscles relax and wrinkles go away. Atropine is an antidote for this, and so we can also have chemicals that can undo the effects of other drugs. Right? And in fact, like soldiers that are deployed in the Middle East, where some of these biological weapons and nerve agents are being used, had syringes of atropine that they carried on them to inject themselves if they felt like they were exposed to a biological weapon. All right. The biogenic amines, this includes dopamine, norepinephrine, epinephrine, that family of all from the tyrosine precursor. Drugs that affect that, cocaine, amphetamines, drugs that treat ADHD, Parkinson's, schizophrenia. Dopamine, as we'll talk about in a minute, is the primary reward neurotransmitter in the nucleus of accumbens. So any drug that acts on dopamine is highly addictive. Serotonin is part of this sort of cluster, although it comes from a different precursor. It is primarily emotion, but also feeding and circadian rhythm states. Some drugs that affect serotonin, I mentioned LSD, Molly or MDMA, and then you have your antidepressants like your selective serotonin reuptake inhibitors and your monoamine oxidase inhibitors. Histamine, mentioned that also today in class. Histamine is an arousal and attention neurotransmitter in the central nervous system. In the peripheral nervous system, it's released in response to inflammation and irritation or tissue damage. Antihistamines and caffeine both act on histamine. Primary breakdown for all of these are typically enzymatic and reuptake. And the monoamine oxidase is for serotonin. The COMT is for dopamine and norepinephrine. Lots of receptor subtypes. So I will say this about this family is that these serotonin and dopamine, let's just talk about those two. Serotonin and dopamine primarily are neuromodulatory, meaning they are acting usually through metabotropic receptors to change the excitability states of other neurons. All right. Serotonin, a little bit more about that. 5-HT, as I mentioned, sleeping, eating, arousal, emotional state. Okay, so one of the reasons that we know that serotonin is involved with emotion and the limbic system is because people that are depressed, when we increase serotonin over time, that we can reduce those symptoms. So that's where this correlation between decreased serotonin levels and major depressive disorder came from, right? The effectiveness of agonistic drugs to decrease depressive symptoms. All right, serotonin comes from the Raphe nucleus, and you can see that it has this diffuse projection throughout all parts of the cortex, the basal ganglia, even the cerebellum, right? Even in the spinal cord. So neuromodulatory, acting in lots of different places in the brain. The medial forebrain bundle is going to the amygdala and to the hippocampus, and supplying serotonin there. And so that's a very important pathway. And then we have to the forebrain, where we think a lot of the limbic emotion systems are affected by serotonin. And then, as I mentioned, throughout other parts of the brain as well, all throughout the brain. In sort of evidence of its neuromodulatory effect, serotonin axon terminals don't have necessarily discrete spine, what we call terminal buttons, where they're matching up to another receptor, like axon terminal to dendrite spine. They tend to sort of be released to bathe the surrounding neurons with serotonin, to have sort of a diffuse effect where they're modulating their excitability. Okay, norepinephrine. Also widespread release, as you can see, throughout the entire brain. Originates from a midbrain nucleus called the locus ceruleus. It's associated with arousal and vigilance. So any stimulant drug is probably increasing norepinephrine in the brain. That's one way that those drugs can produce their stimulating effects. Dopamine. Dopamine also comes from a midbrain nucleus. We actually have two different ones. The ventral tegmental area is supplying the neocortex and the nucleus of accumbens and hippocampus. And this is going to, and primarily going to the frontal lobe. So unlike norepinephrine and serotonin, dopamine is not being released throughout the entire brain, but is pretty much localized to the nucleus of accumbens, frontal lobe, amygdala, hippocampus, and basal ganglia, involved with reward, motivation, planning, and attention. The substantia nigra is a second dopamine release that's being sent to the basal ganglia. It's primary pathway to the basal ganglia, and this is what deteriorates in Parkinson's disease. So we know that dopamine regulation in the basal ganglia is important for proper motor control. All right, nucleus of accumbens. Nucleus of accumbens, since I'm recording this, I can give you a little backstory and tell you a little bit about how the nucleus accumbens was discovered. So, right, I can't point because you can't see, use my mouse. So right here is the hypothalamus. And part of the hypothalamus, a large part of the hypothalamus, is involved with regulating feeding and feeding behaviors. And so some scientists that were interested in feeding behaviors were inserting electrodes into the hypothalamus so that when an animal worked for food pellets, it was electrically stimulating the feeding area of the brain. And so they were inhibiting that through electrical self-stimulation, and then the animal, it was turning off that feeding nucleus. And then they were watching, okay, how hard will that animal work for food when we inhibit this particular hypothalamic area? Well, they put a rat in there and expected it to do its normal thing where it had its effect on feeding. And the rat started pushing the bar and wouldn't stop pushing the bar, just pushing, jamming on it as hard as it could until it passed out from exhaustion. Food pellets piling up behind it, doesn't care at all. It's just self-stimulating. And it would push 2,000 bar presses in an hour until it fell over from exhaustion. It would wake up, straight back to the bar. Wouldn't eat, drink, anything but self-stimulate. The scientists are like, what in the world? We have never seen a rat do this. So they go to see the histology of the brain, right, to see where the electrode was. And it turns out they missed putting the electrode in the hypothalamus, and instead they put it in this area called the nucleus of accumbens that nobody really knew what it did at the time. And the animal was inhibiting the nucleus of accumbens. And so inhibiting the nucleus of accumbens produces a pleasurable feeling. So this animal was just giving itself pleasure by pressing that bar. Research said, great, let's figure out how this nucleus of accumbens works. And so it turns out that dopamine is the inhibitory neurotransmitter that produces the pleasurable feeling, and so that's like the gas pedal, if you will, but it's inhibitory, which is kind of tricky, but the brain does these sort of double negatives a lot. And glutamate from the frontal lobe works in opposition to sort of decrease the pleasure feeling and get back to homeostasis, right? So these two things work together. So any drug that is going to increase the effectiveness of dopamine or decrease the effectiveness of glutamate can activate the nucleus of accumbens pleasure feeling, and that makes it highly addictive. All right. Yes. Okay. So some examples. Amphetamines are dopamine agonists. That's how they produce their pleasure feeling. PCP is a glutamate antagonist, and that's how it has its addictive qualities. Okay. All good. The dopamine, as I mentioned, comes from the ventral tegmental area, which means if we have a drug like nicotine, which acts primarily to increase ventral tegmental activity, then it has a downstream effect of increasing dopamine at the nucleus of accumbens. The frontal cortex is our brakes system, our control, our judgment system. And often what we'll see with addiction is that drugs start to decrease glutamate's effectiveness in the frontal lobe so that it becomes harder to control the nucleus of accumbens. Right? All right. So anything that decreases activity in the frontal lobe is going to remove the brake system from the nucleus of accumbens, and anything that increases activity in the ventral tegmental area is going to increase the gas. Okay. One more specific sort of class of some neurotransmitters. Peptides. I'm not going to really talk a whole lot about peptides in here. There's 5 general classes: brain gut, opioid, pituitary. We will talk about opioids, obviously. Hypothalamic, and then some others. They're typically good for long distance communication, kind of like hormones, so they may be released as hormones. And they tend to be modulatory, right? So substance P is a peptide that is the neurotransmitter of pain and temperature, sensory neurons, and opioid neurons are an inhibitor of substance P. They actually act through that head rib receptor that I just talked about. So endorphins and enkephalins can be released onto the axon terminal of a substance P neuron in the spinal cord to shut down that incoming pain signal. We talk about closing the gate or opening the gate for pain, and that's done through the opioid type of endogenous opioids, endorphins and enkephalins. Synthesis, there's precursor amino acids for these that then get modified by enzymes. They typically are broken down by enzymes. There's lots of types, but again, mainly metabotropic. some peptides can even cross the membrane and have direct effects inside neurons on some of those second messenger systems like the kinases. All right, so here, this is a summary of everything that we've talked about. The last thing that I want to talk about is memory formation, and in particular LTP and LTD, long-term potentiation, learning, long-term depression, decrease in learning. All right, so, and I said that we were going to talk about AMPA and NMDA receptors. So particularly, so the hippocampus primary job is synthesis of long-term memories, right? Converting short-term memories into long-term memories through neural networks in the temporal lobe. So in the hippocampus, glutamate is released in response to something. That glutamate binds to both the AMPA and the NMDA receptors, and the AMPA receptors allow some sodium to come in. We get an EPSP, gets a little more excited, okay? But the NMDA receptor, even though it's opened by glutamate, a magnesium molecule likes to sit and block that NMDA receptor and keep it from doing its thing. If we have repeated stimulation, learning, right, memorizing, reciting something over and over again, right, then that gets encoded into long-term memory. So we have repeated stimulation, we can get enough sodium to come in through the AMPA that it gets positive enough inside that magnesium is no longer attracted to the inside and bumps off, and now calcium can come in through the NMDA channel. And calcium is a signal to that neuron that it should strengthen its connection by putting more AMPA receptors in the membrane. So calcium coming in after magnesium is released is that signal. So, excuse me, we know that calcium activates at least two different kinase pathways, calmodulin and protein kinase C. There are two primary actions that happen after LTP. The first is we're going to increase the number of AMPA receptors present in the postsynaptic side of the synapse. And that's primarily a job for calmodulin. Protein kinase C plays a little bit of a role in that, and expand that receptor density. Second, we're going to release a retrograde signal back from the postsynaptic back to the presynaptic that's going to tell that neuron, hey, I put 5 more AMPA receptors in. You don't have to release as much glutamate in the future because I'm going to be more easily excitable because I added a bunch of receptors, right? Why do we care about this? Because endocannabinoids, AEA, anandamide, and 2-AG are two endogenous chemicals that are released as this retrograde factor, and they go back and bind to CB1 receptors in the central nervous system and CB2 receptors primarily in the peripheral nervous system to decrease future release of neurotransmitters, right? So when somebody smokes marijuana, the delta-9 THC can bind to the CB1 receptors, and it actually impairs memory formation. The endocannabinoids are primarily broken down by enzymes, and as I mentioned, TCH and CBD are two agonists of those. Okay, so long-term potentiation, we are inserting more AMPA channels, right? Because of protein kinase C and calmodulin, right? That increases the excitability. If we don't continue to use this synapse heavily, we can unlearn something or forget it, right? And that's called LT depression, long-term depression, LTD. And there we're actually removing those AMPA receptors that had previously been inserted in there, which makes that synapse harder to function, right? And weakens the strength of that synapse. And so we weaken that neural network or that memory. All right. That's it for today. Appreciate the extra 30 minutes of class time that I was able to get by recording this. We are on track Wednesday to start talking about addiction, and then we were going to start jumping into some different drug classes. So enjoy your Labor Day holiday, and I will see you Wednesday at 11:30. Aloha.